A fraction of a drop of this fluid can trigger complete respiratory collapse. The moment it injects, it begins shutting down the nervous system, rapidly coagulating the blood and destroying surrounding tissue. Yet that exact same lethal fluid is one of the most valuable biological assets on Earth. 1 gram of pure king cobra venom fetches up to $10,000 on the global market, more than 100 times the price of gold. Generating these complex proteins in a laboratory is currently impossible. There is no synthetic shortcut to recreate the molecular structure of snake venom from scratch. To secure this multi-billion-dollar resource, the pharmaceutical industry relies entirely on extracting the venom directly from live, deadly reptiles. This creates the need for a highly specialized, ultra-hazardous profession within herpetology, the snake milker. It is an incredibly small labor force. Worldwide, there are only an estimated 50 to 100 commercial milkers operating today. Those few dozen people act as the absolute bottleneck between global medical demand and nature's deadliest biohazards. The extraction protocol begins with a sterile glass beaker covered in a latex membrane. The snake bites through this barrier, which prevents any saliva or blood from the snake's mouth from contaminating the venom sample. To initiate the bite, the milker grips the snake directly behind the jaw. This immobilizes the head and forces the fangs forward into a striking position over the glass lip. Once the fangs puncture the latex, the milker manually massages the venom glands on the sides of the snake's head to force out the liquid. Every single extraction requires a human worker to physically manipulate an agitated, lethal animal. A single slip of the grip carries severe biological stakes, which generally fall into 2 categories of toxicity. Look at this diagram showing nerve transmission at the cellular level. Cobras and mambas produce neurotoxins, which block these postsynaptic receptors. They sever the chemical messages traveling to the organs, resulting in rapid paralysis and shutting down the brain and lungs. Vipers inject hemotoxins. These destroy red blood cells and trigger massive coagulation collapse. The blood thickens while simultaneously depleting the body's clotting factors, leading to uncontrollable internal bleeding. The manual extraction procedures designed to avoid these outcomes were pioneered in the 1940s by Bill Host. Over his career, he survived over 100 venomous snake bites while handling species like the king cobra. Surviving this daily threat requires adhering to strict sterile protocols written through decades of his trial, error, and near-fatal strikes. Once collected in the beaker, raw liquid venom is highly unstable. Left alone, the proteins will quickly degrade and lose their medical potency. Labs stabilize the liquid through lyophilization, flash freezing the venom and extracting the moisture, leaving behind a crystallized powder. To manufacture antivenom, researchers inject harmless microdoses of this venom powder into large host animals, usually horses. The horse's immune system naturally produces neutralizing antibodies in response. Technicians harvest the blood, purify those specific antibodies, and bottle them into the medical-grade antivenom vials shipped to hospitals worldwide. Through this supply chain, a lethal toxin is processed, filtered, and transformed into a biological shield. But antivenom production alone doesn't account for a $10,000 per gram valuation. The true driver of that price point is cutting-edge pharmaceutical research. Biotech isolates specific destructive venom proteins, reverse engineering them into targeted drugs. Hemotoxins are repurposed to safely thin blood for blood pressure and stroke treatments. Paralyzing neurotoxins are in trials as non-addictive pain relievers and even tested for breaking down cancer cells. The next generation of medical treatments relies on the precise application of nature's most potent biological weapons. And that pipeline only exists because a few individuals are willing to risk their lives to squeeze a snake's fangs over a glass beaker.